Now start the session. So welcome all to the week 12 of Biomass Conversion and Biorefinery. The course coordinator for this course is Professor Kaustubh Mohanty from the Department of Chemical Engineering, IIT Guwahati, and I am Aryan Oja, your MRFTA from the Department of Technology, IIT Roorkee. Currently, I am doing PhD under the guidance of Professor Vibhor Kumar Rastogi from the Department of Technology, IIT Roorkee. Before moving on to the session, first let me share something about myself. I have done my BTech from Bipin Tripathi Kumaon Institute of Technology, Dwarhat, and then later I did MTech from Packaging Technology from IIT Roorkee, and currently pursuing PhD in Paper Technology from IIT Roorkee, and receiving Prime Minister Fellowship from 2021 to present. And currently, I'm working on the project of paper-based value-added products, which is the topic of my PhD program also. So today we will discuss about the assignment 12 questions. Uh, Basically, you all have solved these questions in the previous week. So we'll discuss about the answers, what were the answers, and what is the concept behind those questions. So let's get started. The first question is: ethanol is produced as a main product from which of the biorefineries? So is it a syngas biorefinery, biogas biorefinery, plant platform refinery, or sugar platform refinery? So among these, we know that ethanol is basically we produce ethanol from mostly carbohydrates. Till now, we have learned about this thing that carbohydrates are used to produce ethanol, and from where we get these carbohydrates, we get it from the biomass. So among these, the sugar is a type of carbohydrate. Sugar is a type of carbohydrate. So therefore, the answer is sugar platform refinery. Moreover, sugar platform refinery is used to produce ethanol because we also get some type of bioproducts also, such as uh, syngas. We also can get some type of fuel such as syngas, which can be used to as a power source for this plant itself. We can get we get ethanol also, which is also known as bioethanol, which is the main thing. But to run this whole plant, the production of syngas can be done, which can power the bioethanol. And further, we can get some other types of byproducts also, which can be used in various other categories, which we will discuss in next slides. So, typically, biomass can be utilized to produce bioethanol, which is a type of fuel, and this can be done with the help of sugar platform refinery. So, and moreover, we can get some kind of syngas also, which is also known as clean gas, with the help of thermochemical methods on the biomass. So in this way, we can convert the biomass into the bioethanol and some type of gases also, which is known as syngas, which is a type of power uh, to generate electricity. So in a typical sugar platform refinery, ethanol is typically produced from various types of feedstocks, which are rich in carbohydrates that can be converted into ethanol through a series of biochemical reactions such as enzymatic hydrolysis of cellulose or starch or fermentation of cellulose and distillation resulting in ethanol. So basically what we are doing a biochemical series of biochemical reactions such as we take the biomass, we convert this biomass treat this biomass with some chemicals to take the cellulose out of it, cellulose or starch out of it, and then we convert these cellulose and starch into monomer sugar, and these monomer sugars are then fermented to produce ethanol. So this is done at this part, this process. And secondly, we can get also some other types of materials or chemicals such as syngas by some thermochemical analysis, by treating thermochemically in the absence of oxygen, in presence of oxygen, or by direct combustion, we can produce some type of energy also. So in this way, we can get some other chemicals and energy or materials which are useful products. So therefore, we use sugar platform refinery to produce ethanol and other byproducts. So, ethanol is mainly produced from sugar platform refinery. By far, we have seen this, and sugars. And do we get the sugar? We get the sugar from biomasses. We get these from the biomasses such as corn, sugarcane, or cellulosic material through different pretreatment and enzymatic hydrolysis method. The extracted sugars. These extracted sugar are then fermented using yeast or bacteria to produce ethanol. So we are just treating it to produce some type of carbohydrate, which is done by pre-treatment methods, and then we are uh, doing the depolymerization to produce monomer sugars, and then we are fermenting it with the help of bacteria to produce ethanol. So this is a typical sugar platform refinery process. This process is very similar to the process which we have learned in the previous weeks also, in which we saw about the production of ethanol from molasses, like just like traditional sugar-based ethanol plant. The reason that we are utilizing sugar-based biorefineries when compared to molasses, it is because that in sugar platform refinery we can use any biomass which is carbohydrate rich in in its source. We can use any type of biomass, and it allows for the use of variety of feedstock that can be converted to sugars, which can then be fermented to produce ethanol. And moreover, there is a second advantage of it, and that is that it can produce co-products such as animal feed, organic acids, and lignin, which can be used in various other applications. So, sugar platform refinery not only produces ethanol, but we also can get some other type of co-products also, which can be utilized to produce, which we can produce these materials for other chemical applications. So this is also known as biorefinery approach, that to produce valuable products from biomasses or biomasses waste, which can be utilized in uh, various applications. And however, the production is of low volume. In these products are of low volume. Basically, these are low volume products. We get it in very low volume, but then also we can the thing is that the application is very high, because we get such numbers of such variety of chemicals that we can utilize these all the chemicals anywhere in any type of industry, but the production of volume is low. So this is also known as biorefinery approach. Biorefinery approach means to produce variety of chemicals, variety of chemicals, but in low volume from a single source. So from a single source, if you can produce a variety of chemicals, that too in low volume, then this is known as biorefinery approach. So that's why sugar platform refinery is the procedure to make ethanol, as we can get very type of chemicals which can be used for various types of industries. Now, moving on to the next question. In ICBR technology, which part of the crop was utilized for the production of ethanol? So, first of all, what is this ICBR? ICBR means integrated corn based refinery. So, it first of all, we know that what is it used corn. It uses corn for the production of ethanol. And just wait a minute. So it utilizes corn to produce ethanol. But what does this integrated mean? Integrated means it can use any type of, uh, it can use any part of corn. We know that corn has corn kernels, which is the seeds which we eat. It has a stem, it has leaves, stem, and kernels. So basically, a normal corn-based refinery you only utilize kernels to produce ethanol. But ICBR technology, that is integrated corn based refining, it can use kernels, leaves, and stem, all of it, to produce ethanol. Therefore, the answer is whole crop. That in ICBR technology, we can produce ethanol from the whole crop itself. So uh, basically, it is divided into two uh, pathways. I will explain this in the next slides. So, first one is about corn kernels. So, if we have corn kernels with us, we can just melt down the kernels and to reduce the size. And this, after this size reduction method, we can do the saccharification. Saccharification means nothing but to produce sugar out of it, so that we can get what we say used to say soluble form, to produce soluble form of carbohydrate. This is also known as depolymerization, that we are depolymerizing the carbohydrates present in the corn kernels and to make sugar out of it. As we are making sugar out of it, so we are calling it a saccharification method. So after saccharification, we can do is what is known as fermentation, and this fermentation is used to produce ethanol. And this ethanol can be purified with the help of distillation method, so that we can get high pure ethanol, and this high pure ethanol can be transported. But this first process, is utilizes only corn kernels and not the leaf. It doesn't utilize these leaves or stover or stems. It doesn't utilizes the stover. So to utilize the stover, we just need to add one step, and that is pre-treatment. Why so? Because these stovers have very high amount of hemicellulose and lignin. And to obtain the ethanol from these corn stover, we need to remove this. We need to remove this and produce only cellulose out of it. The stover has all three parts of it, and we need to remove the hemicellulose and lignin and produce only and to get only cellulose. Therefore, one more process comes in between, and that is known as pre-treatment method. And this second part is ICBR technology, that it can utilize any type of uh, any any part of corn, either it is stover or kernels. So what it is doing is, first of all, it is milling just to reduce the size, and then we are doing pre-treatment. This pre-treatment can be done with the help of alkali or acids, or hydrolysis, normal addition of water or steam or any organic solvent. Any type of pre-treatment can be done, which we have studied in the previous weeks also. I think we have studied this in week 3 and 4. So these types of pre-treatment can be applied on the corn stover to remove the hemicellulose and lignin. So hemicellulose and lignins are removed, and lignin is a very sticky material. So it is important to remove lignin because it will inhibit the saccharification process. So lignins and hemicellulose are removed in this pre-treatment method itself. And then after that, when we get the pure cellulose out of it in the pre-treatment method, when we get this pure cellulose, we do the saccharification. Saccharification in the same way as we are doing in earlier also, to produce sugars. And if we have got the sugar, we will again do the fermentation to produce ethanol and then purify it with the help of distillation. So this whole process can be used to produce a bioethanol with the help of whole corn product. We can utilize a corn stover, its stems, its kernels, everything. We can utilize every part of the corn. So this is also known as ICBR technology. In short, we call it ICBR technology, which is a short for integrated corn-based refinery. So, what is ICBR technology? It is a technology that utilizes whole corn plant for the production of ethanol. It can and what whole corn, what are the parts which can it utilize? It can utilize the stalks, cobs, leaves, kernels, and other plant materials. The corn is first harvested and processed to separate the kernels from the rest of the plant, and the kernels are produced using traditional fermentation process, while the other plants are processed using advanced technologies to break down the cellulosic material, which is also known as pre-treatment method. Which and this pre-treatment method is done to remove the hemicellulose and lignin. To remove these hemicellulose and lignin, we do the pre-treatment method, and so we can get cellulosic material. And after this, cellulosic material is converted to simple sugars through a method known as saccharification, which is converted into ethanol by the fermentation process. So theoretically, ICB technology is to produce the ethanol from the whole corn plant. And the reason for using whole corn plant is just that the kernel, if we used only kernel, then the amount of ethanol which we will get, it will be less. So to produce or increase the amount of ethanol, we can take the whole corn plant itself. And corn kernels contain only starch, which can be converted to ethanol using traditional fermentation methods. However, the rest of the plant, including stalks, corn leaves, contains cellulosic material that can be converted into ethanol using advanced technologies such as enzymatic hydrolysis and fermentation. So, in this way, the ethanol yield can be improved per acre of corn, because now we can utilize the same amount of from the same amount of land. If we can utilize the stalks and cobs and leaves of the corn to produce ethanol. So our efficiency also increases, and therefore our yield increases, and therefore we reduce the overall cost of producing ethanol also. So therefore, ICB technology is used to produce ethanol apart from the sugar platform refinery processes. Now, moving on to the next question. What percentage of increase was observed in 2016 in the global production of soybean? So, soybean is mostly produced uh, by USA, and they have made a very high advanced genetics of soybean, which has a very high yield, growth rate, and which we saw particularly in 2016. In 2016, there was a 17% increase of soybean. USA was the highest producer of soybean that time also, and this was done because soybean can also be utilized as a biomass. It can be utilized as a biomass to produce biofuels. Apart from sugars or corn, soybean can also be used. And moreover, we can produce some type of oil also. The soybean oil, which we use for cooking, is the major product of soybean, which is utilized to produce. It is also a type of cooking fuel. So the production increase in 2016 was 17% due to the increasing demand of soybean oil also. So there are several reasons that contributed to the increase of global production of soybean. And one was the favorable weather, and and additionally there was that the soybean products can be used for the animal feed and also produce biofuels, which led to the increase in the cultivation of the crops in many regions. And finally, the advances in agriculture technology and practices such as improved genetics and more efficient fertilization irrigation techniques have also contributed to higher yield. But majorly, the role of soybean in growth was to produce soybean oil for cooking purposes. So now moving on to the next question. What are the steps that are involved in the refining process in an integrated soybean refinery? So integrated soybean biorefinery is a process to produce soybean oil, and it utilizes all these three processes: degumming, deacidification, and clarification. All. But what are these processes and how is soybean oil made? So first of all, degumming. Degumming is the removal of phospholipids from the crude oil. So when we when we crush the soybean seeds and produce the oil, it is in crude form. So the crude oil is then purified with the help by the help of hexane, and the hexane extracts these phospholipids, and thus this is also known as degumming. This can be achieved with the help of physical or chemical methods. After this, the degummed oil is then deacidified because there are some type of fatty acids in the oil. So these fatty acids are not good for health. So deacidification is done for the removal of free fatty acids. So this is also done by physical, chemical, or enzymatic methods. And at last, clarification is done, which is also known as uh, clarification of oil is done, which is also known as bleaching. So we do the bleaching of oil, clarification of oil, or we can say purification of oil. So these three methods are done to produce soybean oil. So and at last there is one more method which was not in the option, and that was deodorization. And deodorization method is done to remove any type of foul smell. So in the options of this question, there were only three options: degumming, deacidification, and clarification. However, a fourth option is also there, that is deodorization. That is to remove the foul smell which is present in the oil. So this is a typical diagram of how do we produce the soybean refined oil from the soybeans. So first of all, we do the crushing, and the crushing is majorly to reduce the size, or crushing of seeds is done, and these crushed seeds are then sent for extraction with hexane. This extraction with hexane is also known as degumming method, degumming process. It is used to remove the phospholipids. There are some types of phospholipids in the soybean, so it is done to remove these phospholipid, and then we get the crude oil. This crude oil is then sent for refining, and then it is further sent for in this refining process. There is a process known as deacidification, which is also known as neutralization. And why it is done? Because we this crude oil is has free fatty acids, and these free fatty acids are not good for health. If we eat these, if we cook our food in free fatty acids, the food can get easily oxidized, and which will be not good for our health. So it is sent for refining, and here deacidification is done. Deacidification means we neutralize these acids, free fatty acids, and then after this, the neutralized oil is sent for bleaching process, and this bleaching is known as clarification. And after this clarification, we get is pure oil without any type of color. And finally, sometimes we also send it for deodorization. And this deodorization is used for removal of some type of odors in the form of any type of gases. And we can get refined oil, which can be used for cooking purposes. So in this way, we get soybean refined oil from soybean seeds by these four processes. That is degumming, deacidification, clarification, and finally deodorization. So this is known as soybean biorefinery. Now let's move on to the fifth question. The major drawback of traditional treatment technologies employed in the valorization of food waste are. So when we valorize the food waste, so first of all, what is valorization? So valorization means to produce any type of valuable product, to produce valuable product from waste. So this is known as valorization. So the question is about food waste. So during the valorization of food waste, that is, if we utilize the food waste such as mango pulp or seeds or any type of this, what are the drawbacks? So the major drawback is the sludge production. Sludge production is the major drawback, and secondly, the ma- other drawback is the emission of greenhouse gases. When we utilize these food products and produce some types of valuable products out of it, during the physical and or chemical processes, some types of gases are produced, such as carbon dioxide or carbon monoxide, and these are released into the air. So the emission of greenhouse gases is the major problem with the valorization techniques. So the emission of greenhouse gases, mainly methane, during the anaerobic digestion process and the generation of large amount of sludge, which requires proper disposal, are the main environmental concerns associated with the traditional food waste. And moreover, the process of traditional food waste treatment can be time consuming. So another thing is that it requires a large amount of time, because food waste do not have any much. It doesn't have a high amount of valuable products. Mostly these are the weights are taken up by the seeds or leaves or some type of extractives. So therefore, the time it is also time consuming. But the major problem with this is the production of sludge and the uh, production of greenhouse gases such as methane. So now the next question is, the internal rate of return is defined as. So it is a internal rate of return is also known as IRR. In short, we call it IRR. It is a question of agri-economics. In agriculture economics, many people uh, learn about it. So what is internal rate of return? Internal rate of return means that at what rate, at what interest rate, or at what rate the net present value, NPV is known as net present value, becomes zero. So what does this mean is that when we do the costing of any type of project, any type of industry, we uh, take out the projections for future. For, let's say, we take out the projection of first year, that how much earning will be there in first year, then in second, then in third, and so on, and until 10th year we are doing projection. And then we will there will be some interest rate also, that let's say the economy is growing by uh, let's say by 10%. So, and if we assume that the earning in the first year will be 10 lakhs, so what is the present value of this 10 lakh after first year? So we will reduce the 10% from this 10 lakhs and take out the present value. And in a similar value manner, we will take out the second, third, and so on. We will take the present value of all the futuristic projections, and at what rate, at, let's say, at X%, at what rate this net value, this will become zero. So when NPV becomes zero, at that, what is the value of R? What is the in, value of R, or also known as interest rate or simply rate? This is known as IRR, internal rate of return. So this is majorly used in economics and not in technical education. But yes, in nowadays everything which we are doing, for example, if the utilization of biomass to produce any type of fuel, we need to be calculative in the we need to calculate the future of this business with the help of the internal rate of return. So therefore, we also need to utilize the economics model in the in these techniques also. The next question was, what is the amount of pectin, hexogens, and pentosans present in citrus waste? So first of all, what are these? What are these pectin, hexogens, and pentosans? So first, these three are type of hemicelluloses. These are hemicelluloses, and the pectin is mostly found in fruits, and apple is the major example. Apple is pectin rich, which gives it a tangy flavor sometimes. And hexosens and pentosans are mostly found in woody or non-woody plants. So the citrus fruit, citrus waste mostly has 25% of pectins, 26% of hexosens, and 7% of lactosans. So basically, the pectin is in the range of 20 to 25, hexosens are in the range of 25 to 30, and pectosans are in the range of 5 to 10%. Therefore, the answer closest is 25% pectin, 26% hexosens, and 7% pectosans. So, pectin, hexosens, and pentosans all are types of hemicelluloses, uh, which is and what is a hemicellulose? It is a complex carbohydrate that is found in the cell walls of plants, along with cellulose and lignin. And hemicellulose is made up of various types of sugar monomers such as xylose, arabinose, mannose, galactose, and glucose, which can vary depending on the type of hemicellulose. And pectin, on the other hand, is a type of hemicellulose that is commonly found in fruits. Pectin is commonly found in fruits and vegetables, while hexosans and pentosans are commonly found in food and other plant-based material. Therefore, whenever we will study about citrus fruits, that the question was about citrus fruit. So pectin will be in higher quantity. It is in higher quantity, that is 20 to 25%. It can be more than these also. But in case of woody or non-woody plants, in case of woody or non-woody plants, the pectin is not a major product and it can be less than 10 or even 5%. So therefore, in fruits, pectin is in higher quantity. What is a typical composition of biohydrine? So first of all, what is this biohydrine? Biohydrine is a combination of gases such as hydrogen and methane and CO2. And these are produced by uh, thermal processes to produce. If we treat the biomass and produce do the gasification of it, we will get uh, methane and hydrogen out of it. And after this, if we do produce the syngas out of it, we will get CO2. So the combination of these two processes will produce these three gases, which is known as biohydrine. So first of all, what is biohydrine? Biohydrine is a gaseous fuel produced by anaerobic digestion of organic materials from food waste or agriculture waste or any type of waste. It is a mixture of hydrogen, methane, and CO2. Biohydrogen is produced from two processes. First is the fermentation process. So the fermentation process can be done on any type of organic material. If we utilize any organic material and do the fermentation on it, we get two types of gases such as CH4 and CO2. So this is also known as biogas. So to produce biogas from the biomass, we can get methane and carbon dioxide. And when we will do the gasification of it, we can get is what is known as syngas. And what is syngas? Syngas is CO plus H2. So combining this hydrogen from here and CH4 and CO2 from here, these three combination is known as bioethane. So, uh, bioethane involves two-step processes. First is the fermentation by bacteria to produce biogas, and the second is the gasification process to produce synthetic gas. So now we can also adjust the amount of methane, carbon dioxide, or hydrogen. We can adjust these composition by the help of controlling the gasification or the fermentation process. So, and by this different types of controlling the process, we can get varying amount of uh, combinations. And biohydrogen is typically 10 to 15% of hydrogen, 50 to 55% of methane, and 30 to 40% of CO2. The amount of hydrogen in bioethane can be increased by adjusting the fermentation and gasification process. So if we just adjust the fermentation process or adjust the gasification condition, we can control the production of hydrogen, and thus we can also control the composition of the gases. So therefore we can produce bioethane. So bioethane has several advantages over traditional fuel gases, such that reduced greenhouse gas emission. So how it is how we are doing the reduction in greenhouse gases? We are producing more hydrogen from the biogas. So biogas has CH4 and CO2, and both are greenhouse gases. But if we do the we do the gasification of this process to produce some type of syngas, we can convert it to produce hydrogen. This CH4 can be utilized to produce hydrogen, and therefore the amount of hydrogen we can increase by controlling the gasification process, and thus we can reduce the CH methane from it. Therefore, we are reducing the greenhouse gases. And moreover, this is a type of renewable production because we are producing it from the waste. We are generally producing it from waste which are organic in nature. Therefore, bioethane has a advantage over fossil fuel gases. It and it can be used for heating, electricity generation, and transportation purposes. So in this way, bioethane has a advantage over the traditional fossil fuels like natural gases. So, the main components responsible for the production of hydrogen from food waste are, so the hydrogen. Now we have seen that food waste, we need some food waste is we need to do the fermentation, and this fermentation we need to do gasification. But this fermentation majorly we require a type of carbohydrate material, and therefore the answer for it is cellulose and starch, because cellulose and starch are rich carbohydrates, and these can be converted to sugars, and these sugars can be easily fermented. And after these fermentation of these sugars, we can get methane and carbon dioxide, and further we can do the gasification to produce hydrogen. Therefore, we need cellulose and starch to produce hydrogen from food waste. So, cellulose and starch are the main components responsible for the production of hydrogen from food waste. And cellulose is a type of carbohydrate found in cell walls of plants, while starch is a carbohydrate found in mainly many foods, including grains, potatoes, and corns. Both the cellulose and starch both can be broken down into simple sugars. We know that we can break down these into simple sugars, which are also known as soluble sugar or soluble carbohydrates, and therefore these can be easily fermented. These can be easily fermented by the help of microorganisms and thus produce biogas. And those biogases can then further be uh, converted to syngas and produce hydrogen. So these biogas can be produced to syngas, and these syngases are combination of hydrogen. So therefore, the cellulose and starches can be utilized to produce hydrogen from food waste. And there's one more method to produce it to produce hydrogen from the food waste is, and that is dark fermentation, which we have studied in the previous weeks also. That we need to, if we have a biomass which is rich in cellulose, we can use to retreat it to produce cellulose and sugars out of it, and these sugars can be used for dark fermentation by some type of bacteria, and thus we can produce hydrogen. So therefore, uh, the biomass, cellulose or carbohydrate-rich biomass, can be utilized to produce hydrogen either by dark fermentation process or through the gasification process, and uh, uh, further doing the any other type of process. Now, moving on to the last question of the assignment 12. What is the first step of performing the financial and economic cost analysis benefit? So, to perform the first step in performing the financial and economic cost analysis method is the identification and description of both the benchmark scenario and the investment scenario. So, what is this benchmark scenario mean? So, benchmark scenario means the method which is already available to us. So, let's say uh, we have a method to producing bioethanol from direct sugarcane. Bioethanol, we can produce bioethanol from sugarcane, and we have every type of every type of result of it. We have the yield of it. We know the cost per kg, cost per land, area of land. We have every type of data for it. So this is known as benchmark. This is known as benchmark data. And if we are producing some other kind of method to produce bioethanol, if we are utilizing some other kind of method to produce bioethanol from, such as from mixed waste, mixed food waste, let's say, and if we do the yield cost analysis per kg per land area, we do everything of this, then we have to compare it with this benchmark, because if it is not coming better than this, then we should not start the uh, other financial cost analysis methods. This should be the first method to compare. So the first step in performing the financial analysis method is to compare with the benchmark scenario and the investment scenario. Therefore, it is the first step to go towards the financial cost analysis. The first step in performing financial and economic cost benefit analysis is the identification and description of both the benchmark scenario and the investment scenario. And this involves defining the current situation or baseline. What is baseline? Means benchmark scenario. What we are considering as the best thing which is available to us, because we will only compare with that thing, and then we will make our own product, and then outlining the proposed investment and its expected impacts. And only after establishing this scenario, if we have established these scenario, if these scenarios are better than the benchmark scenario, then we can go for incremental cost and benefit of the investment to determine and compare the net present value or IRR and the financial metrics. After that, we should do these other financial comparisons. So uh, that's all for today, as this was the uh, last week questions, and further I think that after week 12 now you have your own, uh, you will be having your exams of for NPTEL. So before that, uh, uh, we will also have, we might have one more session, that is week 13, and in that we may discuss the overview of all the topics. We'll discuss the uh, every important topics, a type of summary of the all the week 12s. So hopefully we'll be doing that next week, and we'll see you that time. And if you have any type of questions which you may encounter in the future while preparing, and if you, after the meeting, if you want to contact me, you can always email me. I'm writing my email address in the chat box. I've written my email address in the chat box, and you can always contact me at that email if you have any type of questions in the future. So thank you everyone. Thank you for listening, and I wish you all the best for the exams. Thank you.